Sorry this is so late, I just got home, but I'm going to go over enzymes for the quiz tomorrow. So a catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. So it's just a little helper that helps one thing become another. Like I help you get A's on your bio quizzes. I'm not consumed, but I help you guys. An enzyme is a catalytic protein, so it helps you inside the body speed up reactions. So it's an organic molecule. Like for example, see sucrose is being split apart and by hydrolysis, and the enzyme sucrase is helping with that, and you put it on top of the equation because that means that it's not being consumed, it's just helping. So on top of the arrow you put the name of the catalyst. So the way that enzymes speed up reaction is that they lower the activation energy. So in any reaction, that you have to put in some energy for the reaction to start in. You need to like starting a car, you need to start the car for it to move, and that takes energy. So the enzyme lowers that energy, so it more of it happens with less energy put in. But enzymes don't change how much energy is lost or gained total. You see, like the difference remains the same. It's just that this little hump is lowered. And enzymes are substrate specific. That means that they can only do one process. Like they can't do a bunch of things. It's just one enzyme for one substrate. It's like very good relationship, monogamy. Um, And again, substrate is the reactant in the equation. It's the stuff that becomes the product. It's the stuff that's there before the product. And the substrate attaches to the active site of the enzyme. That means like the part where the substrate fits and that's where the reaction happens. And the enzyme usually changes its shape a little to make sure that the substrate fits. Here you see the substrate coming into the active site and the enzyme changing its shape a little to wrap around it. And here there's a really good diagram. So the substrate comes in to the enzyme and it goes in. The enzyme helps this substrate split apart. The products are released, and now there's an empty spot for a new substrate to go in, and it's just a big circle, and it happens again and again. And again, enzymes are reusable, like I showed before. See, this is reusable because it makes the products go away, and then there's you could use the space for another and substrate. And again, they're very specific. They don't change the reaction equilibrium, so they don't. So like whenever the reaction was gonna stop before because something runs out, it's gonna stop even with the enzyme. It just might stop sooner or later than it would have without the enzyme. And these enzymes can do both the forward and reverse reaction. So in this case, it could split up the molecules, or the two molecules that are the smaller parts, it could go in the enzyme and they can make the big molecule. It can happen both ways. And again, these are just the ways that they lower activation energy. I'm pretty sure we don't need to know like all the specific types because you didn't go over class. Just make sure you know that it lowers activation energy. And if you really want to know, just go over this a little if you have time. And again, since the enzyme is a protein, it depends on its shape for its function. If it loses its shape, it becomes denatured and the protein doesn't work no more. And again, so let's go into the things that influence reaction rates. This is definitely going to be on there because it also was the thing that was on the lab, and that's really important. So if you increase the substrate concentration, there's going to be an increased rate because the enzyme has more stuff to react with. Like in the toothpick lab, if you had 5 toothpicks, or if you had 75 toothpicks, you would run out after a little while. But if let's say you had like 1,000, you would run out after a lot longer. So the reaction keeps on going, and it keeps on going at a higher rate because you don't have to put in the effort to keep on finding new ones because there's so many there. And again, as enzyme concentration increases, the rate increases because if there's more enzyme, the reaction can happen more often, and it's just happy more products. Temperature has a major impact because if it's too cold, the enzymes can't move and they can't do the reaction. But if it's too hot, the enzyme loses its shape because the bonds are broken in the enzyme. Each enzyme has an optimal temperature, like for humans we like 38 degrees, but for some bacteria they really like 80 degrees because they live in hot places. pH also affects it because each enzyme has an optimal pH. Like in your stomach, all the enzymes like really acidic pHs because the stomach is full of acids that break down our food. In our blood, they like neutral pHs because our blood has a neutral pH, so the enzymes work well in that condition. Um, and then like the one that we did in the lab, catalase, it worked well under basic conditions because that's just how it falls under. And you could see that there's like whole range, and if it's in the if it's in its unoptimal pH, the reaction won't happen. Ion concentration also disrupts the also affects the reaction rate because if you have too many ions, then the R groups in the enzymes, they're gonna interact with the ions and it's gonna lose its shape a little because the polar R groups are gonna be attracted to the ions. So it changes its tertiary structure if you remember that from the last chapter. Many enzymes require non-protein helpers like cofactors for catalytic activity, and like usually they're inorganic. Sometimes they're inorganic or they're organic, and the inorganic ones include iron, for example, like hemoglobin. It has a bunch of iron in it, and the iron helps it carry the oxygen. So, like it need like without the cofactors, the enzymes won't work. And then like organic cofactors made out of carbon and hydrogen are called coenzymes, and those that's why we take vitamins so we can get like the perfect amount of coenzymes to make our proteins do their jobs. And this is really important, and it's going to be on the quiz. Um, Inhibitors, they stop the enzyme from working, and there's two main types of inhibitors. If the some inhibitors just instead they're the same shape as the substrate, and they just fall into the active site of the enzyme and they block any substrate from coming in. But this could be knocked out, so it's a competitive inhibitor because it has to compete with the substrate for the for a spot on the enzyme. And if you have a lot of inhibitors, then the substrate will have no chance of getting to the enzyme because all these inhibitors are going to get to it before it. If the inhibitor binds somewhere other than the active site, it blocks substrate binding via non-competitive inhibition. So inhibition. So like this inhibitor, it's gonna go into this little pocket or some other pocket in the enzyme, and that's gonna change its shape a little, and then the substrate won't fit. So this is gonna be permanently there, and it's gonna make the enzyme not work at all. So you can see it's non-competitive because it doesn't have to compete anything for the spot because it changes it permanently. And then this is a really nice summary of it. So more enzyme equals faster reaction rate, more substrate equals faster reaction rate, too high, too low temperature slower, too high, too low pH slower, and with inhibitors it's slower. And then some you can enzymes sometimes it makes too much product, so you have to control whether the enzyme's working or not to keep a balance of the um, substrate and the product in your body. So that's what allosteric regulation is for. Um, It's just a fancy way of saying like turning the enzyme on and off based on what you need. And this picture does a good job of explaining it. So when you want the enzyme to work, this like an activator comes in, and this activator makes changes the shape of the enzyme a little so it works, and so the substrate can go bond and it can do its job. But once there's too much of the product, an inhibitor comes and attaches to the um, enzyme. It changes its shape a little, so it can't work anymore, and so the substrate keeps on accumulating, and there's a lot of substrate, and then the activator makes the substrate work, and then it makes more product, then the inhibitor lets the substrate keep on going, and it's just a big cycle. And then just another form of activation. Sometimes a substrate can attach to an enzyme, and that allows other substrates to attach to the enzyme. It in a way like activates the enzyme because the substrate just gives it a push over the edge, and then it becomes a happy working enzyme. Sometimes and this is not really that important, but this is in feedback inhibition, the end product of a metabolic pathway shuts down the pathway, and is better shown through this example. So this blue thing, thiorein, in the reaction is being converted to isoleucine, and it keeps on doing this. But if you have a lot of isoleucine, it needs to stop making it, right? Otherwise, you have an imbalance. So isoleucine gets attached to the enzyme, and that stops the enzyme from working. And until all the isoleucine is used up by the cell, when the isoleucine is used up by the cell, the it goes away, and then the enzyme goes back to normal, and the reaction happens again. But until the isoleucine gets used, there's no point in making more, so it stops the enzyme from working. And that's feedback inhibition, and that's basically it. That's what you need to know for the quiz tomorrow.